moving on to talk about phylogenetic analysis today, we saw in the last class and in the videos that I posted about biodiversity and microbial diversity. So we saw something about like how much life we have out there, and we tend to kind of like recognize organisms based on what we see on our naked eye, so trees, animals, humans, and everything else. But we do have of the vast majority of the biodiversity, it's actually in form of microbes. Now, how do we study all this life out there? How can we understand the evolutionary relationship among this biodiversity that is out there? That's what we are going to see today with the phylogenetic analysis. So for this unit, or for this chapter, our learning objectives, it is you should be able to understand or to read a phylogeny, to interpret the evolutionary history of organisms using a phylogeny, so know how they are related to each other based on that representation. We should also be able to use genetic data to build a phylogeny and to identify an unknown organism. And that is exactly what the activity for this week will be working on, identifying an unknown organism, constructing this phylogeny based on genetic data, and apply parsimony to choose the best phylogeny. So obviously we need to know what is parsimony and how we can apply it to choose the best phylogeny. So how can we study the history of life, all this life that we saw there that exists based on our biodiversity, all the life we have on the planet? We study the history of life, or when we study the history of life, we are actually doing or studying the phylogeny, or the evolutionary history of a group of organisms. How do we do that? We can use two major tools for that to reconstruct this history of life. First, we can use the fossil records, and this has some pros and cons and has some limitations, for example, because not all organisms that lived on Earth left their fossil records. But we can also use phylogenetic trees, like these ones that I have represented here. So even though they have different topography, different shapes, they actually are trying to do the same thing, which is represent the evolutionary history of this group of organisms that we have here. These two tools, they can be used together or separately, so we can reconstruct the history just based on the fossil records, or we can use the fossil record register to construct a phylogenetic tree. So what exactly it is a phylogenetic tree? It is a graphical summary of this history. So it's a graphical summary of the evolutionary relationship among all these individuals that we are studying. So this phylogenetic tree, it can be constructed based on the relationships among genes, species, and higher taxa. So we can see, for example, instead of having species here, we could have families, we could have orders, or so on. So taxonomy can also be part of this phylogenetic trees. Here's an example of the representation of all the relationships among all living organisms that we have, or at least the most recent representation. So it is what we call the tree of life. That is the most recent version that we have, trying to describe the evolutionary relationships among all living organisms. So we have up here all bacteria, then we have eukaryotes down here, and then archaea. This tree was actually proposed by Laura Hugg in 2016, and then later was updated by Castile and Banfield in 2018 by adding this new group of archaea here. So that is how all life on Earth living today, it is kind of like related to each other. We as a human belong to this group here, or we are one of these small traits that you are seeing here. I like to give this as an example just to show you how much diversity we have out there that we do not even think about. So think that human would be just like one of these small lines here. Look at how much more we have out there. just this big group, and then we tend to think that most of the biodiversity that we see out there, it is what we are actually seeing with a naked eye, and all of those things basically they fall in this category here of eukaryotes as well. So plants and animals, they are all here, but they do not represent most of the biodiversity that we have. Look at how much bacteria we have, how many archaea we have there. So the actual bulk of biodiversity, it is in other clades rather than eukaryotes. But humans are or belong to the eukaryotes, so we are here in the tree of life. Now, why this matters, why we have to study the relationship or the evolutionary relationship of species, things that happened in the past. Why do we care about things that were extinct? Well, because phylogenies, they are useful for many things. For example, we can test hypotheses about evolutionary history. Are we mostly related to chimpanzees? Are we mostly related to gorillas? Things like this. We can use phylogenies to test those hypotheses. We can use it for identifying unknown organisms. And a very good example of that, and very current one, it's what happened with COVID. So remember that the SARS-CoV-19, it was SARS-2, it is a virus, and it belongs to a family of viruses that we had some already. So what scientists did when this emerging pathogen first appeared was to sequence the genome and then match it with the genome of the other viruses that were out there and see where it would fall in that tree. And so we could identify that new organism that appeared. It can also be used in taxonomy to define a species. So we have a very narrow definition of species. Organisms should be able to breed among them. And so we can use this phylogenies also to define what is one species, what organisms are included in one species. It can use in medicine to study the spread of disease. Again, here, another good example using COVID. The new variants appearing there. How can we study the spread of that? How can we know where the new variants are appearing? By constructing trees, sequencing or acquiring genomic data of the virus in a region, and then we can construct a tree and see, okay, so this variant, it's mostly related to the virus that appear in the United States and in Brazil, in India, and so on and so forth. It can also help in identifying species that are a conservation priority. When we have a low genetic diversity inside of that species, that should be one priority in order to conserve that species. It can also be used in agriculture, so for our food, it also matters to identify wild species for breeding with current crop. Hopefully by now you are convinced that it is important to study phylogenetic trees and to know how to kind of like estimate one phylogenetic tree. Now we need to know what is inside a phylogenetic tree, what are the parts that make one phylogenetic tree. First thing that we have, it is our root. So the root of the tree, usually represented like this one here, it means the ancestral lineage. So it is the common ancestor between all the taxa that is represented here in the tree. So if those are species, then this would be the common ancestor between all of those species. It's as to say that back in time, once in a time, all of those species originated from this ancestral lineage here. Then we are going to have the branches. So those lines, in this case here, using this topography of tree, it is a horizontal line. All those lines here, they are branches. So we're gonna have internal branches like this one, and this one. And we also have the branches that go to the tip of the tree that represents the taxa that we are working on. Now, all these branches, you're going to see that they always kind of like start from a node, from this a point or an intersection between these branches here. Nodes inside the tree, they are where the lineage diverge. So here it's also ancestors, but it's not the ancestral one. It's like not the original, the local one. It is the most recent common ancestor. So we're going to have one ancestral lineage, which is like the common ancestor between everyone. And then according to how they are related to each other, they're going to share a most recent common ancestor. For example, node number 2 represented here is the most recent common ancestor between taxon A, B, C, D, E, and F. But taxon A, B, and C shared another most recent common ancestor, which would be located here. The same way taxon B and C would share a most recent common ancestor, which is going to be here. So the nodes represent the most recent common ancestor. Is it different from the root? Yes, because the root is the common ancestor between the whole tree, all the taxa that is represented here. Most of the nodes, they branch off in 2 branches. So like we have here, here, and here. It means like they were the same species or the same population, and then they diverged here, and some group follow one line and another group follow another line of evolution. But in certain cases, we're going to have situations like this where we have more than 2 branches coming out of one single node. This is what we call a polytomy. So poly for several. And what does this represent? It is an unresolved pattern of divergency. In other words, means that we did not have data enough to know if taxon D is most closely related to taxon E or to taxon F, and so on and so forth. So this should still need some more data in order to clarify and make it a divergency like this. So it's just an unresolved pattern of divergency. Now, when we have a species, or I shouldn't say species because this could be family, for example, when we have taxa that it's like this, they share the most recent common ancestor, represented here by number 4, we're gonna say that these two are sister taxa. They are the closest relative to each other in this 3 because they share the most recent common ancestor. In the tips here, when we have the branch that goes until the tips, what we have represented, and we have here randomly just like taxon, this is the group of organisms we are studying. So this can be a living or an extinct species or other taxon. So it can be, like I said, a group of organisms in the same family or different families or orders or any taxonomic level. We can also have genes, for example, here, and it can be living or extinct. That's why we can use fossil records, for example, to estimate a phylogenetic tree, and then we are using both of those tools together here. Now, the other thing that it's missing, it is to name what taxon G represents in this tree. Do you see that we have the root here, which is the ancestral lineage, so the common ancestor between everyone represented in this tree. And then probably something appeared here that diverged 2 groups. One formed taxon G, and the other one gave rise to all the other taxon that we have in the tree. So taxon G diverged from all the other taxon before they diverged from themselves. So it's going to help us to root the tree because it's going to be closely related to the whole group that we are studying, but it diverged before. So it's kind of saying like we had the very first organism that appeared on Earth, and then from there we started branching off and forming life as we know. So our actual group of study would be from taxon A until taxon F. So that is the group we are interested in understanding the relationship among them. Taxon G is here just to help us to root the tree, and that's why we call it an outgroup. Everyone here we're going to call in-group, and then the taxon G, it's going to be outgroup. How you can recognize an outgroup? The outgroup, it's always going to share only the ancestral lineage with the whole other organisms. So it's going to diverge like this always. Now, another small thing that it's missing here, it's how we represent traits, characteristics that it's going to make one taxa different from each other. And usually when you have vertical lines like this, and then you can have numbers, or you can have, let's say, characteristics like jaws, for example, vertical lines inside or crossing the branch, they represent traits. And one trait, it can be present, or if it comes like this and then crossed, means that was lost. That trait was lost in this taxon here instead of acquired in that taxa. But we're gonna see a little bit more about this in a few minutes. Now we know what makes a tree, how we can read that tree. So we need to know how we can estimate, or how can we draw one phylogenetic tree. And to do that, the very first thing we have to do is to construct a data matrix. That data matrix is going to be the basis for our drawing, or for our representation of the relationship of each other. So to construct this data matrix, first thing we have to do is to determine which is the group of organisms we want to study. So let's say I want to compare several different species of plants, or I want to know the relationship between, let's say, all the fishes in a lake, for example. And then once we choose the group of organisms that we are looking to study, so that would be our in-group, we are going to determine which characteristics we are going to use. What do I mean by characteristics? So a characteristic would be a character or a trait, would be any genetic, morphological, physiological, developmental, or behavioral characteristic that we can use to study. So that we can use to distinguish between one organism and another. And when we select those characteristics in the group of organisms we are studying, there is only two possibilities for organisms related to that characteristic. It is either present or absent. So to make it easy, in our data matrix, we are going to represent that by 1 when it's present, or 0 if it is absent. Let's give one example here. So this is the taxa, or this is the group of organisms I want to study the relationship among them. So I have a lamprey, a bass, a frog, turtle, and leopard. I choose the organism. What are the characteristics I'm gonna use to study this relationship among them? Those ones. So I have the backbone, the hinged jaws, 4 walking legs, an amnion, and hair. Now I have to construct a data matrix. So here I have my matrix, and I'm going to start by determining who will have each of those characteristics. So for example, the very first here, backbone. Among these organisms here, I'm going to see who has the backbone. Well, we know that all of them will have a backbone. So I'm going to add 1 for backbone for each of those organisms, because backbone is present in the lamprey, it's present in the bass, in the frog, the turtle, and the leopard. Then the next characteristic is the hinged jaws. So it is this articulated jaws that we also have here. Well, the lamprey does not even has a jaw, so leave alone to be hinged. So the lamprey will get a 0. But everyone else here does have this hinged jaw, this articulated jaw here, so they will all get 1 for this characteristic. And like this, we can do for all the other characteristics. For example, 4 walking legs. We know that the lamprey and bass, they do not even have legs, So they both will get zeros, everyone else will get one. Amnion is less intuitive, but when we study these organisms, we're going to know that only the turtle and the leopard will have. And then the hare finally is going to be just the leopard. Great, so we already have the characters, we have our in-group. Now, the next thing that we have to have in order to have this phylogeny, it is an out-group. So that species or that group that is going to help us to root our tree. So as I said in the other slide, the species or group of species that is closely related to the in-group, so the group of organisms I'm interested in studying, but that diverged before that original group. So in that case, it should have diverged before the lamprey, the bass, the turtle, the frog, and the leopard. Why it is useful to have this outgroup? Because it's going to help us to distinguish ancestral from derived traits. So one ancestral trait, it is a character that existed in one ancestor. In the case when we have ancestral traits in an outgroup, if the trait is present in the outgroup and in the root, means that that was one ancestral trait. In comparison, a derived trait, it is going to be a modified form of the ancestral trait that it's found in the descendant. So it's not going to be present, for example, in the outgroup, or it's not going to be present, for example, in the groups that diverged before that one. And let's see this in a tree so it's easier to kind of like understand. For our example that we are drawing, I choose the lancelet as an outgroup. And so let's see how this lancelet actually falls in this data matrix here. So I put it here, and then we're gonna see which of those characteristics the lancelet will have. So the lancelet, you don't need to know what is this, but it does not have any of those things. It does not have backbone, hinged jaws, walking legs, amnion, or hair. So it's zero for everything here. This is going to be really useful because we know that all these traits, they are going to be derived and not ancestral because they are not present in our root, and it is not present also in the outgroup here. Now, how can we move from having this data matrix to one actual phylogeny? Traits that we choose it here, it doesn't need to be exactly structural traits like this, but it can be morphological like these ones, it can be genetic, or it can be behavior. So different types of data it's used here. And when we use data like that to estimate trees that we are about to create, we can use two kind of like approaches. One, it is going to be what we are going to focus on in this course, which is the cladistic. And the cladistic uses parsimony, and we're going to see what parsimony means in a minute. Or we can use also another approach that it calls phonetic. So we are not going to see what phonetic means or how to construct based on phonetics. So let's focus on cladistic and use cladistic to estimate our phylogenetic tree based on the data matrix that we just constructed. Here we have, so we know that we have lancelet as an outgroup, and it does not have any of the characteristics that we have for the other organisms here. So our root would be here, and we know that our outgroup, it diverged before everyone else that we have in the in-group. And the very first characteristic that appeared was the vertebral column. So vertebral column was not present in the lancelet, but was present in everyone else. So in the lamprey, bass, frog, turtle, and leopard, it does have. So it appeared very early here in this divergence, right after our in-group diverged from the common ancestor, from the ancestral lineage, vertebral column appeared here. Then later, we had hinged jaws appearing here. But our lamprey, it does not have hinged jaws. So it diverged before, and then everyone else continued to evolve having hinged jaws. Next, we had the four walking legs, and our bass, it does not have legs and does not have four walking legs. So it diverged, and then everyone else evolved or continued evolving, having four walking legs. So on and so forth. Then we have the amnion. Then finally, we have here the hair appearing only in the leopard. So in this example, we are saying that traits were appearing, and then based on the presence or absence of that trait, lineages were kind of like diverging from each other, and then we know that those groups will have that trait. But it could happen that instead of appearing, for example, one trait could disappear. It's not the case of amnion, but let's say, I don't know, I don't know an example I could give with this group of organisms, but let's say trait B, it disappeared in turtle. It was present in everyone else until here, but then it disappeared in turtle and leopard. So we would represent it by one vertical line crossed to say that that was a disappearance and not an appearance of the trait. Great, so all of those characteristics that we were using, or not all, some of those characteristics, they are actually called synapomorphies, and that is the core of our cladistic approach. So what is a synapomorphy? It is a trait that is found in two or more taxa. It is present in their most recent common ancestor, but it's missing in more distant ancestors. In other words, will be present just in one group there. We can also call it a shared derived trait. Why shared? Because it is present in two or more taxa. Derived because was not present in more distant ancestors, was just in that most recent common ancestor. Let's see one example here in this tree that we just created. So I can tell you, for example, let's see what color it has, purple. So the purple here for amnion, it is present. Amnion is present in turtle and leopard, which it is this group here. So amnion would be a synapomorphy, and this amnion or this synapomorphy, it is kind of like helping us to group turtle and leopard together. Can you give me another synapomorphy that is represented in this tree? Or another example of synapomorphy? Now, the groups that are identified, or there are the organisms that are grouped by synapomorphies, it is what we call a monophyletic group. What is a monophyletic group? So the monophyletic group, it is a group of organisms that inherited all the traits or that trait from a common ancestor. In other words, they share a synapomorphy. So this most recent common ancestor between turtle and leopard, it did have the amnion. And so turtle and leopard inherited the amnion from this common ancestor. So that's a synapomorphy, and turtle and leopard would be a monophyletic group. So it includes the common ancestor and all the descendants of that common ancestor. The same thing we have here for this common ancestor, where we have between frog, turtle, and leopard. Why this is a monophyletic group? Because for walking legs, which is the synapomorphy, it is present in the common ancestor and all the descendants of this ancestor. Because frog, turtle, and leopard, it is all descendants of this ancestor here. We can also call monophyletic groups lineages, clades. So clades or lineages, they all are the same thing of monophyletic group. And this is kind of like the core of the cladistics approach. We are grouping organisms based on common descendants. So we are looking through traits that was inherited or not from common ancestor. Every time we have something that was not inherited or that was not in that common ancestor, we have a divergence. So that thing kind of like separated before that trait appeared here. So a synapomorphy, it should be present in the most recent common ancestor and in all the descendants. If one of the descendants does not have that trait, that it's not a synapomorphy, and that as a consequence is not a monophyletic group. I'm gonna see in a minute what that will be. Good, so now we know that we group taxa based on their shared characteristics. We use synapomorphies, we use cladistics and everything else. And it looks like, or better, things are looking actually a little bit like simple. You can just compare, oh, do they do not have this characteristic? If they have, they inherited it from a common ancestor, and that's it, so on and so forth. Actually, as everything else in life, things are not so simple like this, and some possible complications exist when we are constructing a tree. And one of those complications is what we call a homoplasy. So a homoplasy, on the contrary of one synapomorphy, it is a trait that is similar, it's present in two taxa, but it is not inherited by a common ancestor. It is actually inherited by convergent evolution. So the evolution of that trait was independent on the two lineages that will have that trait. So when we have traits that are inherited from a common ancestor, that is one homology. So homo for same, logy, it's going to be origin. So it has the same ancestor. Homoplasy means homo, the same, but plasy, it is for form. It has the same form, but it's not the same ancestral. It was not inherited from the common ancestor. It can be a reversal in a character state, and it can create the appearance that there was no change at all. So again, to account for losses here in this case. Why this is a complication in estimating the trees? Because you can have a species in one monophyletic group when you are comparing trait A, but it's going to be not in a monophyletic group, in another different group, if you are comparing trait B or if you are considering trait B. Let's see how this works in an actual tree. So this is an ichthyosaur. It has, and it's one extinct species, and it has this streamlined body like the dolphins will have. So dolphins and ichthyosaurs, they share the same characteristic, which it is this streamlined body. But the most recent common ancestor between dolphins and ichthyosaurs, it's back here, so it's very far. And the most recent common ancestor between these two taxa did not have that streamlined body. How do I know that? Because the other descendants, for example, the primates, the rodents, the dinosaurs, lizards, monotremes, and everyone else, they do not have that trait. Means that the trait probably appeared here in dolphins and also here in the ichthyosaurs separated. What it made that thing to appear? Convergent evolution. How do we call the group between dolphins and ichthyosaurs? It's going to be a polyphyletic group. So poly, several, phyletic, phylogeny or ancestral. So several ancestors gave rise to that group. We are just grouping it based on that characteristic that they have in common, but that characteristic was not inherited from their most recent common ancestor. So what it means to be inherited or to appear from convergent evolution. Convergent evolution is the process where organisms were submitted to the same pressures in the environment, so they had to evolve similar traits to be adapted to that environment, or the environment um, selected similar characteristics, but was not that they shared a common ancestor. So I like to say that they were separated things because they were in similar places, they were converging to the same thing. They are converging to the same characteristic, but they started in different points. They are going to the same direction, starting from different points. While the synapomorphism, it started and went in the same direction. So it started in a common ancestor and it kind of like branched in all the places where they had the same thing as well. So here's one example of homoplasies: the wings of bats, birds, and insects. Why this is a homoplasy? Because the most recent common ancestor between bats, birds, and insects did not have wings. So why do they have wings? Because all of them had to fly, so they had to evolve this characteristic, but their common ancestor did not have it, was not inherited from that common ancestor. Great. So how can we distinguish between them? So homology, it is the same ancestry, so same logy origin. Homoplasy is the same form. There is a slight difference between homology and synapomorphy, but we are not going to go into this level of details here. Just assume that a homology, it is inherited from a common ancestor. So homology traits occurs when they are traits are similar due to shared ancestry. So we inherited that from a common ancestor. One example of homology, it is the structure of our arms. So the bones that will make the arms of humans, the paws of cats, this I forgot the name of the thing of the whale, and then the wing of the bat. All these bones, even though they have different shapes and they have different function, they were inherited from a common ancestor. So this is a homology. It's a structural homology. And what is the homoplasy? Was the traits are similar because of other reasons, because of convergent evolution, so they are not from common ancestry. And one example, we already saw the wings of bats, birds, and insects. They do not share a common ancestor that will have wings, means that those wings evolved separated more than once in that tree if you are comparing all of them together. So your book will have this picture here, which has some examples of trees, and it also has the definition of different groups that are formed based on the characteristics, either synapomorphism or homoplasies. So we already know that when we have a synapomorphy or a homology, we're going to have a monophyletic group. That trait will be present in the common ancestor and all the descendants. We already know what is a homoplasy. It is a characteristic shared between organisms that was not due to common ancestry, but convergent evolution. And when we have a homoplasy, we are talking about a polyphyletic group because we have several origins for that characteristic. It appears more than one time. And when I have a characteristic, and that's the last one that was missing to say here, when I have a characteristic that it's present in the common ancestor, it's present in most of the descendants, but it's lost in one of those descendants or in a few descendants, that makes for a paraphyletic group. And again, and now here, C, it's not anymore a synapomorphy. Why it's not a synapomorphy? Because it's not present in all the descendants. It's missing in this descendant here. So if we miss one descendant, that is not a monophyletic group anymore, that it's not a synapomorph. The trait is not a synapomorphy anymore. And again, a loss of a trait can be useful in determining divergences in a tree here. So have a look in this table in your book and be familiar with these monophyletic groups, polyphyletic, paraphyletic, and what are the characteristics that we need in order to define them. That could be very useful. Now, this thing that we just made here of constructing this tree based on the characteristics that we attributed in that data matrix, it's what we did was just to use maximum parsimony. So what means parsimony? First, when we are trying to estimate the relationship among organisms, we cannot say, especially if it is extinct ones, we cannot say that for sure that it's how they are related. We are just trying to find the best explanation for the relationship among them. So we narrow down the possibilities by applying the maximum parsimony. What is parsimony? Parsimony, it is the principle that is going to assume that the least number of chains occurred in the path of evolution. So I like to say that parsimony, it is how to say, it's to say that evolution is kind of like very lazy, and if it has to take it's going to take the smallest number of chains possible to have or to explain the organisms as we have today. So if I have to change a trait and then to lose that trait, that's probably less possible than if I just appear that trait once in a tree. So like, for example, in this example here, we had this pulley shaped bone here, this ankle bone. That's the trait. If we have these organisms organized in a tree like this, we're gonna say that the ankle bone appeared once here and then the whale diverged before. So it's out of the artiodactyls group here. Now, if the whale it's actually related to the hippo and it's inside this tree, we're gonna have the ankle bone appearing here once and then disappearing here. So we had two chains occurring. One, it appeared and then it disappeared. When we compare these two possibilities, we're gonna say the first one is most parsimonious because it takes less change to occur in order to explain the relationship among of those organisms. So we probably, what is probably true, it is the tree number one, it's what represents those organisms instead of tree number two, because we have less change needed in this tree. So how can we choose the best tree when we have options, when we have a lot of different possibilities? So we're gonna assume that the tree that requires the fewest evolutionary events, it is the most likely one. So it's when we do that, we are assuming that common ancestry, it's much more common to happen, it's much more probable than convergent evolution. So the same characteristic to evolve more than one time exactly in the same way, it's kind of like much harder than you evolve it once and then you pass that along to the other species. And when we are using few species like the ones that we did in the example here in class, we can do that by hand. But in reality, when we are actually estimating phylogenetic trees, we use computer programs to compare those possibilities and to choose for us which is the most parsimonious tree that we have. We all obviously have other strategies. For example, we can use maximum likelihood. We can use many other different models to estimate or to choose which would be the best tree to explain the relationship among those organisms using the data that we have. Let's see one example here on how to choose the most parsimonious tree, because that is one of the activities you have to do also for the assignment of this week. So here I have what I have as taxa. It is actually DNA sequences that I have here. And this is how those sequences are related to each other. I have the same sequence represented here and here, just that these two trees are representing these sequences in different relationships. For example, here I have 2 and 3 being sister species, and in this case, they are not sister species. What are the changes that appeared in 3A? Well, everyone was the same until this third A, adenine, it actually changed to a cytosine here. And so sequence 2 and 3 was grouped together because they were the only two ones that will have that, until it appeared another change here, the C that changed to a G, grouped 4 and 5. In the 3 number B, what it's saying it is, we had sequence 1, then it changed A to C in sequence 2, then it changed C to G in sequence 3, then it changed again A to C, the same change that occurred here, and it changed again here in the number 5, the same change that occurred in the 3. So when we have these two possibilities, which of those 3 is we going to say that it's most parsimonious? A, right? Because we only have 2 changes occurring here to explain how they are related. On the other hand, here we would have to have 4 different chains, and we would say that on sequence 2 and 4, the exactly same change should have happened in different times in the evolution. So this is less probable to happen than the situation here. So A would be the most parsimonious tree in this case here. So as I said, when we are estimating one phylogenetic tree, that it's not 100% sure, we are just testing hypotheses to see which ones is going to fit or which relationships best fit that data. That's what it's saying here. We narrow down the possibilities using this principle of maximum parsimony, which is going to assume this laziness of the evolution, that it's going to take the fewest events possible to explain the relationship of all the organisms. So with this, we actually finish what I have to review about phylogenetic trees with you.